So hello everyone, in this video we'll talk about the Lac Operon. So first we discuss what is the operon. So operon is a group of structurally gene having a single promoter. It is a structure of operon. So it contains two parts. First is a regulatory part and second is a structurally activated part. These regulatory parts contain a regulatory gene which produce the regulatory protein. This regulatory protein is a regulator this structurally activated group of genes. So now operon has two types. First is a inducible operon and the second is a repressible operon. In inducible operon, is by default in switch off condition. But when signal molecule is come, this signal molecule is going to bind the repressor protein, and this repressor protein is getting deactivated and removed from the operator. And finally, this operon is getting switched on in condition. Now, remember that example of inducible operon is Lac Operon. Lac Operon is the example of inducible operon. Okay. And it's also called the positive regulation of operon. Second, repressible operon. Repressible operon is by default in a switch on condition. But when signal molecule is come, this signal molecule is activate the repressor protein, and this repressor protein is going to bind the operator and switch off the operon. And example of this repressible operon is TRP operon, TRP, tryptophan operon. Which called a negative regulation, or a repressible operon is also called a negative regulation. Now we discuss the Lac operon, which is example of inducible operon, and it is example of positive regulation of operon. So this is the structure of Lac operon having two parts. The first is a regulatory part, or it is having gene Lac I, which called the protein repressor protein, and it has a second part. It has a structurally activated parts which codes protein beta galactosidase, permease, and transacetylase. So now we discuss about the function of enzyme. So repressor protein. What is the function of repressor protein? So it's bind to operator of operon for regulation. Now then beta galactosidase. Beta galactosidase it's convert lactose into allolactose, and second it convert lactose into glucose plus galactose. Okay. Then permease is it's help to entry of lactose or glucose or any other molecule from outside to inside in cell. Then we discuss about the transacetylase enzyme. Transacetylase enzyme is mainly help in the transfer of acetyl group from the acetyl CoA to beta galactosidase enzyme. Okay. So now we discuss the regulation of Lac Operon in a different condition. So when lactose is present in a media, lactose is come to the cell using a permease enzyme. Now this lactose is converted into allolactose using a enzyme beta galactosidase. Now this allolactose is acting like an inducer. So this allolactose is now acting like an inducer because it induced the Lac Operon. Because it induced the Lac Operon. So how it's induce the Lac Operon? Let's see. So now this yellow lactose is going to bind the repressor by two ways. First is bind to repressor having freely moving in cytoplasm, and the second is bind to repressor which is already binded to the operator of the Lac Operon. Now, due to binding of this allolactose, and allolactose is an inducible molecule for this operon, this repressor protein is getting deactivated and this removed from the operator. So now this RNA polymerase is getting activated and transcribe the gene of the Lac Operon, and this called a switch on condition of the Lac Operon. Now here one thing you must be remember that even without lactose, some amount of enzyme is always present in a cell of bacteria. Okay, in bacterial cell. So what is tells that? What is tells that? It tells that even whatever lactose is present or lactose is not present, this enzyme is always present in bacterial cell. Okay, some amount, little amount. So now in second condition, when the glucose is present in media, the glucose is come to the cell using a permease enzyme. Then this glucose is reduce the level of CMP in the cell. Due to reduce the level of CMP in the cell, CMP reduce the level of also CAP. CAP called the catabolic activator protein. So these catabolic activator proteins very helpful in activation of RNA polymerase. But due to here the reduction of the catabolic activator protein, it does not activate the RNA polymerase. Okay. So this RNA polymerase is inactivate condition. So it does not transcribe the gene. It does not transcribe the gene, and ultimately this operon is switched off in condition. So now in third condition or third situation, when a glucose concentration is low in media, the glucose is entered to cell using a permease enzyme. Now, due to low concentration of this glucose, is increase the level of cyclic AMP. Now, increase the level of cyclic AMP leads to the increase the amount of catabolic activator protein in cell. Now, this activated catabolic activator protein or activated CAP is going to RNA polymerase, and this polymerase is activate and transcribe the gene of the Lac Operon. Now here one thing is remember that this type of regulation is called the positive regulation of Lac Operon. Okay, because it's getting switch on condition of the operon. So I am not discuss the mutation here. You just read this table, but you must be remember some words that constitutive expression means this operon is constitutively expressed. Then non-inducible means this operon is switch off in condition or does not express any protein. Then transaction means mutation in a protein called a transaction. And the cis action means here mutation in a DNA sequence. So this is a question which might be useful for your examination. You read this whole question. This is a This is a reference which I used to make this video. And finally, thank you for watching my video. See you again later.